So it's now my pleasure to introduce Dr. Kurt Harris to give us the distinguished lecture on targets for cancer prevention. Dr. Harris, you have to listen to this also. I'm sorry. Kurt is the chief of the Laboratory of Human Carcinogenesis and chief of its Molecular Genetics and Carcinogenesis section at the National Cancer Institute. He's also a clinical professor of medicine and oncology at Georgetown. Dr. Harris received his undergraduate degree from Kansas University and his medical degree from Kansas University School of Medicine in Kansas City. His clinical training in internal medicine was completed at the University of California at Los Angeles and at the National Cancer Institute. Dr. Harris has a wide range of scientific interests and accomplishments spanning molecular genetics of human cancer to molecular epidemiology of human cancer. His scientific contributions to the fields of molecular carcinogenesis and molecular epidemiology of human cancer have placed him at the international forefront. His research on environmental carcinogenesis, cancer risk factors, and molecular genetics of human carcinogenesis, P53 and microRNA pathways has significantly impacted the field of cancer risk assessment and our understanding of the molecular pathogenesis of human cancer. Dr. Harris has received numerous honors throughout his distinguished career, including the Alton Oechsner Award relating smoking and health from the American College of Physicians, the Deichmann Award from the International Union of Toxicology, the Charles Heidelberg Award from the International Society of Gastroenterologic Carcinogenesis, and the Distinguished Service Medal, which is the highest honor from the U.S. Public Health Service, the NCI Outstanding Mentor Award, and the AACR Princess Takamatsu Award. He has published more than 500 journal articles, 100 book chapters, 10 books, and holds more than 25 patents owned by the U.S. government. And in the time that I have discussed this, another paper could have been written. So he also serves as editor-in-chief of the journal Carcinogenesis and has held or currently holds elected offices in scholarly societies and nonprofit foundations, including the AACR, the International Society of Differentiation, the Keystone Symposia on Molecular and Cellular Biology, and the Aspen Cancer Conference. So it now gives me great pleasure to ask Dr. Harris to come here to give us the distinguished lecture on targets for cancer prevention entitled, Inflammation and Cancer, Interweaving microRNA, Innate Immunity, and P53.
Thanks, Eva. It's a pleasure to be here, and the first part of my presentation, I hope, will be complementary to Liz's elegant presentation, which will focus primarily on senescence and P53, and then I'll move into microRNAs and inflammatory genes. So these are some examples of genes and mechanisms that regulate senescence. The major tumor suppressors, P53, which I'll speak mostly about, but also RB, cyclin-dependent kinase inhibitors that are in some cases either downstream or upstream from P53 and RB, such as P21, WAF, or P16. Stress signaling, in this case, I'll emphasize free radicals, both nitric oxide and oxygen-based radicals, and pro-inflammatory signaling, such as IL-6 and IL-8. So P53 has many functions. One of its functions is to induce senescence, and cellular senescence is, in fact, an important tumor suppressor mechanism in vivo. This is from association studies initially, that precancerous lesions and benign tumors contain more senescent cells than do cancers. And secondly, in the animal studies, that P53 restoration can lead to tumor regression through senescence. Studies done by Tyler Jacks and Steve and Scott Lowe and others. And as you've heard, that senescence is an important part of aging and also tissue regeneration or lack of. This more or less emphasizes what Liz was just talking about. Telomere shortening can lead to activation of the P53 pathway and also increase in being 4A. So The first part of the actual presentation is going to be work done by Martin, Kari, and Izumi. And the hypothesis that they've been investigating is that p53 isoforms and microRNAs are physiological regulators of replicating cellular senescence, both in vitro and in vivo. We got interested in this when it was discovered that there were multiple isoforms of p53, some due to a alternate splicing mechanism. In fact, there are now 14 different isoforms of p53, such as p53 beta, in which the carboxy terminus and the N-terminus are alternately spliced in this case, or in the delta p53, you have deletion of the first 133 amino acids because there's a second promoter for the p53 gene that's in intron 4. This isoform of p53 acts primarily as a co-transactivator for wild-type p53, and this isoform acts primarily as a dominant negative form of p53 by binding and inactivating some aspects of p53 function. But there's a possibility that there's gain of function also of the delta 133. So the first very simple experiment was to take human fibroblasts that were either young and growing or those that had reached a senescent state, so Y and S, and these are two different strains of human fibroblasts. And we found that as the cells became senescent, there was an increased expression, stability of the message and protein of p53 beta. There was no change in delta 40 p53, so we put that aside. But interestingly, we found that the dominant negative form of p53, the delta 133, was highly expressed in young growing cells, but was undetectable as the cells became senescent. And the senescent cells had the morphology changes, had phosphorylation of p53 at serine 15, an increase in MP21 protein levels. Now, the most physiological experiment that we could think of was to knock down the delta p53, the 133, using siRNAs at this point. You can see the knockdown is reasonable. This is with one antibody, this is with another antibody, and when the cells had knocked down delta 133, there was an increase in p21 WAF in both of these situations, and an increase in senescence-associated beta-gal activity here and here, and this is the quantitation there. Now, what happens if you overexpress either delta 133 or p53 beta? And I will say that in cancers, generally, delta 133 is highly overexpressed. You get increased growth with delta 133 compared to the control, and the p53 beta overexpression, less growth. And this is associated with an increase in senescence with the p53 beta overexpression compared to the delta 133. And with the delta 133 expression, you actually delay senescence. You increase population doublings. So these are cells that were near senescence, and overexpression of delta 133 gives about another 10 to 12 population doublings. That's quite similar to what you would get if you knock down p53 by itself. And as you can see here, the control, you see increase in p21, but you don't see that when you have the delta 133 overexpression. Now, I said earlier that there's association studies in which in benign tumors you see much more senescent cells and markers of senescence compared to carcinomas. So we analyzed adenocarcinomas, human colon adenomas, and you can see there's an increase in the senescence-associated beta-gal, this low magnification, higher magnification compared to the controls, and an increase in p16 in the adenomas. In the adenomas, there was a decrease in the delta 133 and an increase in the p53 beta. Then we analyzed adenocarcinomas, those that had wild-type p53 and those that had mutant p53. And these are colon carcinomas. So this is the non-matched control for the non-cancerous region in these surgical specimens. And in the carcinoma, there was an increase in the dominant negative form of p53 and a decrease in the p53 beta, the co-trans activator of p53, in the wild-type p53 tumors. But this was not the case in the carcinomas that had mutant p53, in that the delta 133 was unchanged, even though there was a decrease in the p53 beta, the alternately spliced form co-trans activator of p53. So these results are consistent with the delta 133 p53 functioning as a dominant negative. So this goes back to the cartoon of the colon adenoma and the fact that there are both p53 and RB tumor suppressor networks that are senescent barriers to cancer. So induction mechanisms include increase in p53 beta, delta 133, decrease, and an increase in p16. In the malignant tumors, you see a decrease in the p53 beta, an increase in the delta. p53 mutations can eliminate the need, perhaps, for the increase in the dominant negative form of p53, and DNA methylation and transcriptional silencing of p16 can take place, or mutations. Now, interestingly, there are other examples in which free radicals will cause induction of senescence, and oncogene expression, such as RAS oncogene overexpression, will lead to senescence. And that appears to be related in part to WT1, the Wilms tumor 1 gene, that when there is a decrease in that gene expression, that RAS will lead to senescence. When it's intact, when the WT1 is intact, the cells have an increased growth rate. IL-6 and IL-8, which I'll say something more about in a few minutes, are quite interesting because they will also cause senescence. But if this barrier is breached, they act as autocrine growth factors. Now let's turn to the second part of the presentation, which will deal with microRNAs and inflammatory cytokines as cancer biomarkers of risk, prevention, screening are the important issues there, and prognosis, to treat or not to treat. And of course, there are biomarkers that are important for screening for recurrences, drug response, and pharmacodynamics. So let's first deal with microRNAs. MicroRNAs were discovered in C. elegans by Victor Amoros and others in 1993. A rather recent finding, and it's a recent finding because this is a mechanism that goes all the way back to plants. So from an evolutionary standpoint, microRNAs is an extremely old mechanism of gene and message stability and protein translation. The second point is that the output of not one or two, but hundreds of genes are altered by microRNAs. And they do this primarily by destabilizing the message that they bind to, as opposed to inhibiting translation, which was initially thought. In 2000, microRNAs were discovered in human cells. And in 2002, Carlo Croce and colleagues found that microRNAs were differentially expressed in human cancers, in this case, chronic lymphocytic leukemia. This seminal study led to many investigators, including us, to look at microRNA signatures and asking the question, can they predict diagnosis, prognosis, and therapeutic outcome? So the experimental approach that we and many other people use is do a microRNA expression profiling using microarrays, validate that with real-time PCR, and get better quantitation. Do in situ hybridization to ask the question where in the tissue and in the tumors there's expression of the microRNA. Do functional analysis using overexpression or knocking down microRNAs or using anti-microRNAs. And then identification of target coding gene of specific microRNAs by computational analysis. I will say that the current algorithms for predicting which microRNAs target which mRNAs, about only 1/4 of those targets actually prove to be valid with experimentation. Now, what regulates microRNAs? Gene amplification, gene deletion, different kinds of cellular stress, epigenetic mechanisms such as DNA methylation and transcriptional silencing that Peter Jones made the first observation on, and then also inflammation. Downstream from microRNAs, there are multiple targets in multiple pathways, and they function not by altering just a single message and its protein, but by altering multiple messages in multiple pathways. So one needs to think in a systems biology way as opposed to a antisense kind of idea in regards to microRNAs. Now, microRNAs are processed primarily by Pol II to form prior microRNAs. P53 plays a role here by coupling with a RNA helicase called P68 and bringing that helicase to Dorcher, which is a nuclease, and facilitating the opening up of the microRNA to form the pre-microRNAs that are then transported to the cytoplasm by exportin 5 and RAN, and then interacting mostly with dicer, another RNA nuclease. And this RNA nuclease is transcriptionally regulated by P63, which is the ancestor gene of P53. And then, as I said earlier, about 80% of the activity of microRNAs is primarily message stability, and about 20% is at the translational repression. Now, what happens if there's a mutation in P53? One is that you do not get this processing help here. And secondly, some mutants actually bind to and inhibit the P63 that inhibits its ability then to upregulate dicer. So there's decreased processing that takes place. What is the effect of SNPs in P53 and in P63 genes? That's currently under investigation in several laboratories. And then what is the effect of microRNA processing and microRNA gene SNPs? And that's the next 2 or 3 slides. So in processing, there are a number of SNPs that have been identified and their function is being investigated. And a lot of this data and information is in a review that we wrote recently. In the pri RNA, there are SNPs, and one of the most interesting one is right here, associated with MIR15 and MIR16, which is associated with familial chronic lymphocytic leukemia that the Croce lab discovered. And interestingly, in the New Zealand black mouse that has an increase in lymphoproliferative diseases and leukemias, exactly the same SNP is found So this ties together a human disease with the mouse disease. Now, there are SNPs also in the pre-microRNA. One is associated with increased risk of esophageal cancer with alternate RNA splicing, microRNA splicing. And there are SNPs that lead to changes in the seed region of a microRNA. That's about 7 base pairs, which is the most important aspect of the microRNA, but not the only one, that allows it to bind to messages, primarily the 3' UTR. And in this case, this SNP leads to a change in the targets of this particular microRNA. So you get different targets here versus here, and it's associated with increased risk of prostate cancer and breast cancer. And then lastly, and actually the first that was discovered, were SNPs in the message regulatory regions, primarily 3' UTRs, such as here. And Frank Slack and his colleagues, Joanne Wiedhaus, found that in the message of the KRAS gene, there was a SNP that led to inhibition of LET7 binding to and causing genomic instability, message instability, and this is associated with increased risk of lung cancer. And this has been replicated by several other studies. This is such a new area that most of the other examples that I mentioned have not been replicated yet. Now let's go to chronic inflammation and infection and related to cancer. And there are a couple of sessions that will be held during this meeting on this topic. But it's been long recognized that chronic infection and chronic inflammation can increase cancer risk. This can be largely inherited, such as hemochromatosis and increased risk of liver cancer in this iron overload disease, but much more frequently acquired by infection with microorganisms, such as viruses, hepatitis B, hepatitis C, bacterial infections, Helicobacter, parasitic infections such as liver fluke, and less appreciated in the past, but coming to the forefront, are chemical, physical, and metabolic conditions that lead to chronic inflammation, including acid reflux and Barrett's esophagus and esophageal cancer, obesity, which there's a session on obesity and by John Milner and colleagues that is associated with increased cancer risk, and of course smoking. In addition to the 60 or so chemical carcinogens in tobacco smoke, it's also a highly inflammatory agent. The World Cancer Report a few years ago, edited by Bernie Stewart and Paul Kleihuis, estimated that about 18% of human cancers, over 1.5 million per year, are related to infection. And then if you add these to that, I'm sure it's substantially more than 18%. I don't want to leave you with the impression that all chronic inflammatory diseases are associated with increased cancer risk. Rheumatoid arthritis may have a slight increase in association with certain types of lymphomas, but certainly there's not an increase in joint sarcomas. And there are oncogenic viruses, such as human papillomavirus, which itself is not highly inflammatory. Now, it was recognized a few years ago that obesity is a chronic inflammatory disease that increases the risk of cardiovascular disease, neurological disease, and of course cancer. And when people examined fat, they found not only adipocytes and preadipocytes, but they found many activated macrophages and feedback loops that were associated with chronic inflammation. There's also an increase in free radicals, both oxygen-based free radicals and nitrogen-based free radicals, that can directly damage protein and inhibit repair in capases, and also directly damage DNA, and if it's not repaired by base excision repair, can lead to mutation. But there's indirect damage, including changes in DNA methylation and epigenetic changes, but also lipid peroxidation, producing these highly reactive electrophiles, malondialdehyde and 4-hydroxynonenal, and activation of the arachidonic acid cascade. Now, some years ago, we investigated the role of nitric oxide, and we're still, in fact, pursuing this, and showed that, certain cytokines, including interleukin-1 beta and TNF-alpha, will increase the inducible form of nitric oxide synthase for the NOS2 that will produce nitric oxide. This will produce some DNA damage, activate p53 with the anti-carcinogenic effects, and also include induction of senescence. This DNA damage can cause mutations in certain genes, and p53 is one of those that the frequency of p53 mutant alleles is increased in chronic inflammatory diseases such as inflammatory bowel disease, for instance. This can lead to pro-carcinogenic effects, genomic instability, decreased cell cycle checkpoints, and decreased senescence. And you lose this negative feedback loop here, and you get substantial amounts of nitric oxide, and this was first studied by Stefan Alms and Kathy Forrester in our group. Now let's turn to inflammatory cytokines. And I'll show you just one example of some data that's unpublished but is very consistent with lots of other data showing that there's an association between interleukin-6 and interleukin-8 serum levels and lung cancer risk. And this is two different studies, a case control study, population-based controls, and the prostate, lung, colon, ovary prospective study that you're all familiar with, the PLCO study. So serum IL-6 was analyzed, was found to be elevated above the median in the case control study and also in the PLCO study. Interleukin-8 was also increased in both studies. There was a difference, though, that if you analyzed in the PLCO study serum samples that were 2 years or less prior to the individual developing lung cancer, that IL-6 was quite high. However, if you looked past 2 years prior to developing cancer, even up to 10 years prior to developing cancer, IL-8 was predictive of increased risk. And I might also say that IL-6 is also, we published this, and others have too, is associated with survival of lung cancer. So high IL-6, poor survival. Now, there are many studies that have looked at a panoply of inflammatory-related genes, both pro and anti-inflammatory. And again, as I said, Andy Dannenberg and others will be talking about this later in this meeting. These are a couple of studies from our laboratory. One of hepatocellular carcinoma that Xin Wang is the leader on, and then a lung adenocarcinoma, in which there were a number of different inflammatory-related genes that were analyzed, developing a signature, and showing that those that had the signature, primarily a pro-inflammatory state, had poorer survival than those that didn't. And this is also true in lung cancer, although the genes were somewhat different, but many of them were similar. This is all kinds of cancer, and this is only stage 1 cancer. So this is predictive of or associated with the poor survival in those groups. More recently, we've analyzed genes related, inflammatory-related genes, association with colon cancer prognosis. This is a study done by Jane and by Aaron, and the inflammatory risk score in the tumor and non-tumor correlates with colon cancer-specific mortality independent of tumor stage. And these are some of the genes in the non-tumor region of the colon, including an increase in IL-10 that was associated with a subsequent increase in lymph node metastases. And in the tumor itself, an increase in IL-23 that stimulates Th17 cells that release IL-17, which generally leads to increase in metastases. Now, in this study, and I'll show you this slide just to make one point, and that it's very important to have at least 2 cohorts when you're doing these studies. And we tried to choose cohorts which were dramatically different from each other. So this is a cohort from Hong Kong, which we broke into 2 different groups, a training group and a test group. And the high inflammatory risk score was associated with poor prognosis. And then we validated that with a cohort from the Baltimore, Maryland area and got the same kind of result. The next kind of study that we've been doing is combining biomarkers. And I think this may have some importance in biomarkers for prevention also. So the hypothesis was that the combination of inflammatory risk score and miR-21 expression is a better prognostic classifier than either alone. So these are essentially the inflammatory risk score associated with survival, 100% survival, 0%. This is a 5-year point. We previously published in JAMA a study showing that there's high levels of miR-21 expression in colon cancers that are associated with poor prognosis, and that's shown here. But we put the 2 together, we found that if both were low, quite good prognosis. If either one was high, intermediate prognosis. If both were high, quite poor prognosis. And then putting this to statistical analysis, particularly multivariate analysis, to look at multiple covariants, we found that each of these, the inflammatory risk score, miR-21, and tumor stage, were independent of each other. So the next question is, what's the principle here? And the proof of this principle is that the combination of miR-21 expression and inflammatory gene biomarkers improves prognostic classification. And you get curves like this in what I just showed you. What might be the reason for this? Each biomarker properly classifies some of the patients, but they misclassify others. This biomarker also properly classifies some of the patients, misclassifies others, but they're different people. So the 2 together gives you a more robust prognostic classifier. If you put 3 together, is that better than 2? 3 biomarkers that are mechanistically and statistically independent of each other. The answer is yes. If you put 4 together, we don't know the answer to that yet. Now let's end up with a little more discussion of miR-21 and why we and many other people have been interested in that. In a study that we did with the Croce laboratory, looking at 6 major types of human cancer, we found that overexpression of miR-21 was in common in all 6 of these cancer types. And now miR-21 is upregulated in over 13 major types of cancer. So it's the winner so far in terms of the race of which microRNA is most dysregulated, in this case, upregulated. That led us to analyze miR-21 and other microRNAs in lung cancer, and this is a picture of Nazumo when he was in the laboratory, and we found that the profiles were significantly different between primary lung cancers and corresponding non-cancerous tissue among the different histological types, and that these 4 microRNAs, these 3 were upregulated and that was downregulated, were associated with diagnosis and prognosis, including early stage lung cancer. Now, Takahashi and Frank Slack had previously shown that LET7 is downregulated in lung cancer, and again, LET7, among other things, targets KRAS. Now we've looked at 3 different cohorts. The Maryland cohort, that was the initial investigation, a Norway cohort, and a Japanese cohort. Kaplan-Meier plots, 100%, 0%, 5 years. So high levels of miR-21, the upper quartile, poor prognosis, poor prognosis, poor prognosis. This was found in frozen tissue. We've done a similar study in paraffin-embedded tissue, and we did not find a significant difference. And in my opinion, the methods for isolating microRNAs from paraffin-embedded tissues, formalin-fixed paraffin-embedded tissues, still need substantial improvement. And if you look at stage 1 lung cancer, and we're interested primarily in the earliest stages of lung cancer, one can see in the Maryland, Norway cohort combined poor prognosis, and that was correspond also to poor prognosis in the Japanese cohort, which was primarily initially made up of stage 1 lung cancers. Now, is lung cancer the only kind of cancer that's associated with poor prognosis when miR-21 is overexpressed? Well, I mentioned colon cancer previously, but pancreatic cancer, breast cancer, tongue, and astrocytomas have been recently looked at. And high expression of miR-21 in these cancers is also associated with poor prognosis. And lastly, is miR-21 an oncogene? And secondly, is it a prevention and therapeutic target? Well, the first evidence that was an oncogene was in a mouse study that was just published by Frank Schlack, in which he made transgenic mice with a TET off system, miR-21, and if TET off system was activated by doxycycline, the mice lived. If miR-21 was expressed, the mice died. And they died with pre-B-cell lymphomas. How about the other side of this coin? If you already have tumors, such as this mouse, and you knock off by using dox to the TET off system, what happens to the tumor? It gets smaller and smaller and smaller, and the mice live. So this is consistent with a oncogene addiction that Bernie Weinstein, the late Bernie Weinstein, came up with that hypothesis a number of years ago. Well, those are lymphomas. How about carcinomas? So this is a study that was from the Olson laboratory, in which they had transgenic mice that expressed miR-21 in lung cells, and using the Tyler-Jacks KRAS model, they found that there were many more tumors if miR-21 was overexpressed. And if they used the miR-21 knockout mice married to the KRAS mice, they got fewer numbers of tumors. Lastly, there are a lot of preclinical studies in which anti-miR-21 have been used, primarily with human tumor xenografts, and this is just an example of one of those studies showing there's less tumor growth and less cell proliferation in those that have been treated in vivo through the anti-miR-21. So what regulates miR-21? Gene amplification that occurs in many kinds of cancers. Transcriptional silencing will inhibit it, as shown by the Crochet Laboratory. Our group showed that EGFR pathway, including mutations in EGFR found in never smoker lung cancer cases, markedly increased miR-21. The Baltimore Laboratory found that IL-6 through STAT3 increases miR-21. FOXO3A actually inhibits it, and various kinds of stress, including ionizing radiation, hydroxyl radical, and we've also have unpublished data now with nitrogen-based radicals. Downstream from miR-21, again, there are perhaps hundreds of targets, but these are some which are tumor suppressors and anti-metastases genes whose products play a role in inhibiting metalloproteases, tumor growth, focal adhesion kinases, anchorage independent and migration, that miR-21 actually inhibits these. But most recently, miR-21 has been shown to target a mismatch repair gene and its message and decrease MSH2, which leads to microsatellite instability due to a lack or diminishing amount of mismatch repair. So that's the presentation. What's the implications of microRNAs, inflammatory cytokines, and p53 isoforms? Well, insight in signaling pathways and carcinogenesis and tumor progression, and many of us are interested in that. I think that they have a role in prevention of cancer, and it will be to the ingenuity and intelligence of all of you to put that into practice. That diagnosis of cancer, that's actually not so interesting to me except in early stage cancer, and prognosis, particularly of early stage cancers, prediction of therapeutic outcome, and then finally that microRNAs, inflammatory cytokines, and p53 isoforms can be prevention and therapeutic targets, at least potentially. And lastly, I'd like to once again thank my colleagues at the NCI, and we've had the privilege of collaborating with a number of other folks and who've taught me a great deal. I appreciate that. I appreciate your attention today.